Good evening, everyone. I'd like to welcome you all back for the most awaited talk of the evening. And Professor Satyajit needs no introduction, but it would be my honor and pleasure to introduce you to one of the most distinguished scientists of our time, Professor Satyajit Mehr. Professor Mehr, commonly referred as Jitu by his friends and colleague, is a renowned cell biologist at NCBS. He's been the former director of NCBS and InSTEM. His academic career has been multidisciplinary, where he moved after doing his chemistry from IIT Bombay. After that, which he obtained his PhD in in life sciences at Rockefeller University in New York, working in the laboratory of molecular parasitology. After which he moved to Department of Pathology at Columbia University, New York, where he did his postdocs. At NCBS, his lab works in the area of cellular organization and signaling. He has taken a multidisciplinary approach combining cell biology with physics and chemistry to study the organization and endocytic trafficking of transmembrane and lipid-anchored protein in members of living cells. Professor Mehr's work has been published in numerous high-impact scientific journals and has received numerous awards. To mention a few, he has been awarded the Infosys Prize for Life Sciences in year 2012. He's also awarded the Shanti Sarup Bhatnagar Award, which is the most prestigious award, scientific awards in India. But Professor Mehr's contribution to science go far beyond his individual research accomplishments. He's a passionate advocate of scientific education and outreach, working tirelessly to promote scientific literacy and engage the public in scientific process. He has served as numerous scientific on numerous scientific advisory boards, and he has been a mentor and a role model for countless students and early career scientists. So please join me in welcoming Dr. Professor Satyajit Mehr, a visionary scientist and a true leader in his field. Sir, please.
Very kind introduction. And also, it's really a privilege to be here and talk to you about some of my work. But before that, I also want to thank Vinay for being so persuasive and so accommodating of all the various constraints of my schedule. But as of today, I'm happy to say that I have not as many constraints, because I just stepped down as the director, the center director at NCBS, and one of the alumni from this campus, faculty alumni, has taken over. So, I'm sure we wish Sashi well, and so do I, and that he allows me to continue to work for a while at NCBS. So let me start by saying that, given that it's the Founders' Day, I thought I should say a few words about Dr. Bhargava and his sort of extraordinary accomplishments in a time when molecular biology was not really a household word. I mean, today you can ask somebody what is the CT value of your something, and you will get an answer. You can go to somebody on the street and they will tell you that their PCR was positive or negative. But that's all molecular biology. And Professor Bhargava set up CCMB from 1984 till 87. I guess it was transiting between across the road, or that side or this side, I'm not sure which. But and then it became a full-fledged campus in 87. And he, along with somebody who I've deeply admired, Ubaid, set up NCBS, a few years later. And Ubaid and he, I must say, were two contemporaries who, they knew each other well, and there were many exchanges between them, and they had different styles. But their commitment to building science as a fundamental research enterprise was unflinching. And I think this is something that characterized some of these institution builders of the time in the early 80s and the 90s. Today we have a very different outlook. and a very different demand on science. And I think it's important for us to also think about what that means. And there are no such tall leaders who can, who can say, the emperor has no clothes, or whatever the phrase may be to the powers that are in place. But I should add that, Bhargava was a man who never feared to speak his mind and fought hard for bringing about a scientific temper. And in many ways, Obaid did the same, but in a different style. Bhargava was more demonstrative, and he's also famous for having had the courage of his convictions and returning a Padma Bhushan because he felt that there was a very strong climate of fear. And the country was moving against rationality, against reason, and against scientific temper. And this prompted a lot of consternation amongst many. And I must say, many scientists at that time did not come forward and support him. And I think that was not an appropriate thing, because what he was saying is what we are seeing today, more and more of obscurantism in science, and this is not going to take us very far. In fact, science itself will be under threat if we don't take up cudgels, that were shown, and courage that was shown by Bhargava at that time. But I just wanted to say, he didn't do this only at this time. He did have a vision, and I stumbled across a letter, and this happens to be a letter in the archive of the MS Swaminathan papers that the archives at NCBS acquired very recently. It was a letter, in fact, a copy of a letter that he had sent to Rajiv Gandhi, the Prime Minister, in 1989. This is long before the human genome was resolved. And what he did say was that he outlined limitless possibilities if India could get into building up capacity for the human genome. He laid out why it would be a cheap thing to do in India, because scientists were cheap, and they are cheap today too. But he laid out the possibilities that if he could build up the capacity for human genome, unraveling the human genome, we would be at the forefront of molecular biology at that time. He also understood that there was a competitive geopolitical situation, no different from what we are at today. And he had an international perspective. He said the international community would take up cudgels on our behalf if he actually completed this project in the time that he knew it would take, and he provided a way to do that. And he said this would also make us self-reliant. I mean, none of the words are different from what is being bandied about today, but this was being done with a deep conviction that you could actually do something serious at that time. He also then went on to say, I believe that India should seriously consider setting up its own project for mapping the human genome in the 8th plan period with the spade work done during this year. And he would be delighted that CCMB would be the site where this should be located. Of course, we would have thought about that, but this is what somebody who founded this place said, and I must salute the prescience of the man at that time. Anyway, so that said, I want to talk about some of the science that I have been doing, and I hope it may not be as entertaining as anecdotes, but I hope the science will be entertaining. So what I'm going to tell you about is, in fact, what the edge of a cell looks like. It looks and behaves like a fabric, and I want to describe this fabric to you in detail. And the only way I can do that is by taking you into a journey in time, and also a journey about the ideas about the cell membrane. It is also about how two disciplines interact, how physics, in fact, meets biology and often scratches its head because biology doesn't behave, and what the outcome of that interaction is. So that's essentially what I'm going to try and talk to you about in the next 2 or 3 hours. Right? So the yeah, before because at the end of 3 hours I'll forget to acknowledge my colleagues. But let me say, this work is an incredibly exciting multidisciplinary collaboration with so many of my dear friends and colleagues. In particular, theorist Madan Rao and chemist Ram Vishwakarma and his colleague Parvinder, and our students who'd work jointly between us. And I'm going to name people along the way, so I will not at this moment shout out their specific names. But as you can see, it's really, everyone seems to have a smile. And that's something that really excites me. Even Abrar, I mean, that's how he smiles. Yeah. So the eukaryotic cell membrane, this is a cell that looks, rather straggly in a culture dish. It's a multi-component fluid lipid bilayer. That's what the membrane of a cell is supposed to look like. And here's a picture from a dear friend of ours, also somebody who Amit in the audience knows well, Mike Edidin. It's a picture that he drew in 2003, collected over many years, of what a fluid mosaic to look like. But even to get to this picture took over 100 years. And here's the history of how we got to that picture of a fluid mosaic. So and I encapsulated for you very quickly in one slide. The first encounter with physics was, of course, when Agnes Pockels, who was working in her own home. In fact, she developed a way of measuring the surface area of a lipid bilayer, and which then Langmuir made into a much more sophisticated Langmuir trough. But she and he figured out how you could measure the surface area of a single lipid molecule. And then, based on their work, Gorter and Grendel decided to take red blood cells, extract lipids, and then spread out the lipids onto one of the devices that they had made, and discovered that the surface area of the lipids that they had identified was exactly double the surface area you'd expect for the size of the red blood cells and the number of red blood cells that they had identified, or that they had isolated. So they said, well, it must be a bilayer. And in fact, that's where the term the bilayer shows up from. And the bilayer was then sort of in some ways attributed to Gorter and Grendel. But just imagine if they had actually looked not at red blood cells, but at reticulocytes or at some other cell, we would have had a very different picture of the cell membrane. I mean, it was simply because they picked red blood cells, which happened to have no organelles inside them, that they came up with this idea at that time. Then proteins became to be proteins. Proteins were discovered in membranes, because if you precipitated the lipid extract with bicarbonates, you would find proteins in that gamish, and proteins had to be placed somewhere, and there were this trilamellar model where proteins were incorporated in this way. But then in 72, almost 51 years to date almost, Singer and Nicolson proposed the fluid mosaic model, which we still consider as the current, which I will explain to you must change by the end of the talk, the current model for understanding how a membrane of a cell is supposed to be considered or even imagined. And it's about how membrane proteins are distributed in the bilayer, and that's the model from the famous paper of the fluid mosaic, where proteins are embedded in a sort of a sea of uniformly distributed lipids. Already this set of observations over a period of 50 years reveals an intertwined relationship between concepts borrowed from physical studies on membranes, because much of the concepts that produced this model were from biophysical measurements of mobility of molecules, including capping proteins and all that on cell surfaces. And on artificial membranes, connecting that to artificial membrane measurements and cell membranes. Okay, so So what do you have? You have then the fluid mosaic model. Of course, you have lipids, and this is chemistry. So you have several phospholipids. Here are 4 types of phospholipids. That's phosphatidylserine, phosphatidylethanolamine, choline, and phosphatidyl'inositol. And one has sphingomyelin, which is a ceramide backbone, and glycolipids, which are also ceramide backbone lipids. And one has another enigmatic molecule, the sterol, cholesterol. So these are all components of the lipid bilayer. And how does one begin to understand? And first, that's the 5 classes, minimally 5 classes of molecules of lipids in the membrane. So the question then is, where are these molecules coming from? Of course, to understand where they come from, one needs to, ask a question, and the only question one can ask in biology is in the light of evolution, right? So where do these lipids come from? So here is an evolutionary tree of life from circa 2011, where you have the ancestral original cell, bacteria, archaea, and eukarya. And I mean, at this audience, I don't need to talk about the evolutionary tree of life, but this is what we understood about 10 years ago. And then, I must say this, even though the tree of life spans a huge length of time, our understanding of this has changed dramatically in the last few years. The reason, okay, let me start, say, if we had looked at this tree of life, we would have had one problem. Bacteria and so this tree of life predicts that bacteria and eukarya are really far away, and you have to go through archaea, or archaea branches off early, and in fact eukarya are somewhere around here. And so these systems, the archaea and eukarya, must share almost all the enzymatic machinery that arise, that build this structure. Now, they of course share the ribosomal machinery, the DNA replication machinery, but lipids is a different story. The lipids have completely different chirality compared to the archaea. So archaeal lipids are built with a distinct and different chirality, and at the end of the day, the archaea have mainly ether-bonded lipids, whereas in the lipids that are built in bacteria and eukarya, almost exclusively, we have the acyl lipids, distinctly different chirality. So clearly, these two things could not have come from the same. So eukarya, the lipids of eukarya could not have come from the archaea. 2015, loci archaea were discovered, and then the tree of life changes. It becomes now the bacteria and archaea now together contributing to eukaryota. And this is a change because this Asgard set of archaeal species were discovered in some hot vents in the North Sea. And now it becomes clear that you could have contributions of these 2 elements into the building up of the archaea. You could have the DNA replication machinery, ribosome machinery, and the lipid synthesis machinery coming from bacteria, because both of them contribute to the last common ancestor of the eukaryotes. So mystery at least resolved because of the discovery of the loci archaea in 2015. Again, by deep sequencing, and something that perhaps also could have happened here. So, so we have the revised evolutionary tree of life with phospholipids as a focus. Phospholipids, fatty acyl ethers, and isoprenoids, all of them explained by this wonderfully modified tree of life. But then, what about sterols? So you have sterols in some bacteria. You actually don't have sterols. You have something called hopinoids and sterols in eukaryota. And this is an example of climate change. Because at the birth of Earth, the partial pressure of oxygen was incredibly low. And then as time moves on, this is billions of years, the partial pressure of oxygen begins to increase. And it is somewhere around 2 billion years ago that the pathways of sterol biosynthesis emerge because there's sufficient oxygen available for all the reactions to build this really amazing molecule, that's sterols, which allow, if you will, the cells of a bacteria, of a eukaryotic membrane to do all the wonderful things that it does. In fact, that's just the enzymatic pathway where I show that you need 11 molecules of oxygen to make 1 molecule of sterol, and that is what results in the production of these cyclized sterol molecules that contribute to the membrane. And then there's a beautiful statement from Conrad Bloch, who actually identified sterols and their synthetic pathways. He says, selection pressures rendered the sterol molecule progressively more and more, and ultimately optimally competent for membrane function. Now, that obviously is an evolutionary argument. And then he argues that the sterol pathway terminates with cholesterol, a molecule designed to optimize attractive van der Waals interactions with phospholipid chains in the membrane bilayer. And I think this statement, provides an evolutionary argument for much of what I'm going to tell you about the next encounter with the physics of the membrane that we will see now. So what I've shown you now is that the Singer-Nicolson model has this sea of lipids, proteins embedded in it, and we have now some evolutionary sense of where these lipids are coming from. And of course, we now understand that this massive climate change induced by those small creatures, the bacteria, producing oxygen, generated the capacity to make sterols. Creatures like us are doing other things to the planet today, and who knows what will happen next. Perhaps we'll lose the sterols, but we'll have to see. So the second encounter was this idea that cholesterol could pack with the lipids of the membrane and generate very distinct phases in the bilayer. So if you take a lipid bilayer which has lipids above its melting temperature, it is present in a liquid crystalline or liquid disordered state. Below its melting temperature, it usually is present in a solid or a gel-like phase. These solid or gel-like phases are not good because they don't allow mobility, and they are, as the word goes, gel-like. But cholesterol now fluidizes this gel-like phase and creates a liquid ordered phase, which was sort of famously claimed to be the raft phase. So the second encounter of physics and biology at the cell surface was the understanding that artificial membranes, if they have lipids, those lipids can exist in these two different states, because when you take lipids and make an artificial membrane, imbue it with cholesterol, you can see liquid disordered and liquid ordered phases in the membrane, and those ordered disordered phases are thermodynamic transitions of the structure of the bilayer. So they promote sort of liquid phase separation in the bilayer. And this idea now propelled many cell biologists, Kai Siemens, who was a proponent of this idea, to say that in the trafficking system, in the signaling system, membrane lipids would segregate because they have the potential to build ordered domains due to the presence of these distinct types of lipid compositions. And these sorts of ordered domains would then attract other molecules that would prefer to partition into these ordered domains, and now you would build a structure that was capable of collecting signaling molecules and having them, in one place, signaling, or in one place to be sorted to another place inside the cell, because they could be collected by this sort of physical property of being able to partition into these ordered phases. That was a very nice idea, and many of us pursued this question for many years. And of course, we also discovered along the way that the membranes of the cell had very distinct compositions. The cell surface had a particular composition, and each one of them was tightly regulated. The cell surface had a particular composition shown up here. You don't need to look at the different, just look at the patterns. The patterns of the lipid species look quite distinct, going from the ER to the plasma membrane to the mitochondria that we heard about this afternoon to endosomes to, in fact, the Golgi. I mean, all these patterns look very different. And they look pretty much the same if you go from different cell types to cell types. And each cell type also has a very different composition, which is tightly maintained. And this, again, buttressed the idea that you had different phases in different types of cell membranes. But often us biologists tend to be a little naive, and one should always remember the cell is a bit smarter. Yeah? Because the cell happens to live at a temperature where none of that matters. If you take lipids of the composition that I'd shown you and put it into a cell or put it into a membrane and sort of heat it up to the temperature at which the cell, physiological temperature of the cell, whatever be that cell, the membranes are not going to phase separate. The membranes will be well mixed in a happily well mixed thermodynamically stable state. So the cell obviously does not want their membranes to phase separate, right? And but yet, this idea that molecules could partition into these ordered phases started gaining currency, and nobody wanted to consider this idea that the membranes themselves, the cells themselves may not want that to happen because of the temperature business that I just described to you. So what was going on? And it was at this time that I was starting my lab in Bangalore, and and one of the favorite molecules that partitioned into all these different ordered phases was a lipid-anchored protein called the GPI-anchored protein. And so when I was starting my lab in Bangalore, now 96, 95, we put explorers into the membrane of a living cell, introduced fluorescently tagged GPI-anchored molecules, which was supposed to partition into these ordered domains in the membrane of a living cell, and decided to ask the question to the protein itself, well, do you partition into these sorts of membrane domains or not? But to ask that question, well, we in fact already knew the answer, because the images that I've been showing you here of cells are in fact cells that have been labeled by fluorescently tagged GPI-anchored molecules. Those membranes don't look as if they have, in fact, if you take a much closer look, they don't look as if they have these large phase-segregated patches in the bilayer. So, but there could be other structures present in the membrane, and so we had at that time, this is just about the time fluorescently tagged proteins, genetically tagged fluorescent molecules were being invented. We tagged our GPI-anchored proteins with it was a folate receptor. Folate receptor, by the way, is the main molecule that brings in folates into our body or into our cells. That happens to be GPI-anchored. And I had synthesized a fluorescein tagged folate, which I knew bound very well to this receptor, and studied the surface distribution of that molecule using a property of fluorophore that allows one to look at fret between like fluorophores. I had to use like fluorophores because I didn't know how to make a red fluorescently tagged folate. In fact, I tried a lot of time to make another colored folate, but failed miserably. The tags at that time, fluorescent tags at that time were not amenable for the folate to keep its property of binding its receptor for whatever reason. We now know the reason, but at that time we only had one single color, and I wanted to study short-range interactions of these molecules in the membrane. And we used a technique called fluorescence emission anisotropy measurements, where you can monitor fret between like fluorophores, so it's called homofret. And you excite a fluorophore, in this method, you excite a fluorophore, and here I show you a GFP for convenience, excite a fluorophore with polarized light, and if that fluorescence is emitted by this GFP molecule during the time it takes to rotate, the fluorescence of that GFP molecule will depolarize slowly because the protein molecule rotates slowly compared to the lifetime of the fluorophore. And then the fluorophore, if there is a neighbor that is also has the dye attached to it, the fluorophore will actually transfer energy to the neighbor, and the information about the polarization in its initial state is completely washed out. So by monitoring the loss of fluorescence emission anisotropy, one could actually monitor whether there was fret taking place between two like fluorophores or not. And so I'm going to show you what we saw. And we literally, when I say we, it was Rajat, my student at that time, and he saw this picture of the membrane of a cell labeled with this fluorescent folate, and it was very uninteresting, flat, no membrane raft. So of course he was very disappointed. But then he looked at a little patch in the fret channel, and what he saw literally, made him jump out of his skin. So what you see here are extremely depolarized patches in the membrane, abutting extremely polarized patches in the membrane, and this variegated pattern of fluorescence anisotropy distribution told us that the folate receptor must be forming extremely tight clusters here in these blue patches because the fluorescence is highly depolarized. And in these yellow patches, it must be almost sitting as a monomer. So the membrane of a cell was doing something, but at this very short length scale. And the distribution of the fluorophore is, paradoxically highly uniform, right? So what is this? So using, at that time, an understanding of how you could do perturbation fret measurements, we discovered that what the cell was doing was building tiny little nanoclusters of these GPI-anchored molecules in the membrane at a length scale that was actually sort of similar to that of its Foster's radius, which is on the order of about a few nanometers. So this, I'll argue, is one of the first, if you will, super resolution images of a fluorescent molecule in a membrane, at 1998. And then those studies were then followed up by electron microscopy, where people, and not our group, but other groups, John Hancock and Rob Parton's group showed that you could look at this by EM if you did some tricks, and then later on by near-field scanning optical microscopy, and now super resolution imaging from Jennifer Lippincott-Schwartz's lab. All of them confirmed that these GPI-anchored molecules in membranes of garden variety fibroblasts form these tiny little nano aggregates. Okay, so given that, we knew that we then, described the sort of what one would do as a, reasonable physical chemist, describe the clustering of this molecule, its kinetics, show what it was sensitive to. We showed that it was independent of concentration, very odd. The fluorophores themselves were immobile when they formed clusters. It was sensitive to cholesterol, the same molecule that we have been discussing. And if you removed cholesterol, these clusters fell apart, and the clusters themselves rapidly remodeled. I mean, it's very hard to reconcile all these observations in one fell swoop. And to add to that, when the cells make blebs like this, so when the cell makes blebs, those clusters, disappear from the blebs. And what we realized that these blebs were actually detaching the membrane from the cortex, which to us suggested that the clustering that is present at this short length scale, these tiny little nanoclusters, they must depend on an active actin cytoskeleton, because they're not present on the blebs, and when the blebs come back onto the side, attaching to the cytoskeleton, they reappear. So we, of course, were very excited by this, and we already knew that the membrane of any eukaryotic cell is draped over this beautiful fenestrated cortex, where the membrane interacts with the cortex and is pinned down by fences in the cortex, and these fences in the cortex have a specific impediment for the mobility of many molecules in the membrane, including lipids. But that's a passive effect of the influence of this cytoskeletal mesh that underpins the membrane of a living cell. So that's what we knew at that time. And how could the clusters actually form from a structure like this? And so I should say this is when the third encounter with physics started. And I think, to me, this has been the most important one, at least from my perspective of understanding the membrane, where Madan and his colleagues, completely free of any constraint of experiment or anything, only constrained by imagination, said that if the membrane is templated by a dynamic cortical actin, then one way that these clusters could form, and we could find all the properties of how the clusters could be built, if one suggested that there's a contractile active composite template at the membrane, and this composite is sandwiched between cortical actin mesh and the plasma membrane, and the composite consists of actin molecules in red and myosin molecules in yellow. And what happens to these molecules? So they actually built a rigorous theoretical framework which had two key elements, which had no proof at that time from any experiment. That there were short dynamic filaments of actin embedded in this static mesh, and that myosin motor activity was driving these short dynamic filaments to form sort of interesting structures. And applying sort of the fundamental equations of a new area of physics that was emerging, that of the non-equilibrium physics of active matter, and applying sort of the understanding that was coming from that area, which also, by the way, describes the flocking of birds or the shoaling of fish, all of which from the same principles, at least the same equations. And in our case, of course, it was the shoaling of clusters in the membrane of living cells. And I mean, I won't go into these equations in great detail here, but I'm really happy to chat with people after the talk if you would like to understand this in more detail. But sort of more pictorially, that's the model up there. This is the idea that the actomyosin contractility clusters the actin, and if anything is associated with the actin, those molecules are also going to be clustered, right? And the properties that they predicted mapped so beautifully to the quantitative measurements that we had made of the clustering of these molecules. And we could make quantitative measurements because, as I said, from a physical chemistry perspective, that's what we had already determined. So the model recapitulated the key features of the cell experiments and also made certain predictions. First, it said if dynamic filaments are advected by myosin, they should indeed drive membrane protein organization. And then direct coupling to actin should also drive clustering. So we made model proteins that had actin binding motifs at their cytoplasmic tails, and then connected these, expressed these proteins in cells and asked if you could actually find whether these molecules also clustered. And of course they did. So these deep blue patches are extremely highly clustered regions of these transmembrane proteins which have actin binding motifs. And if you get rid of the actin binding motif, or if you get rid of the actomyosin there, these clusters fall apart. They also exhibit active signatures of this concentration-driven clustering, which are fluctuations, which I'm at the moment not going to go into in detail, but they map so beautifully to the theoretical predictions that I must show it to you here. That that's the prediction is in these lines, in dotted lines. That's the experiment of the slopes of the log of the fluctuation versus the log of the average number of molecules in the membrane. They map so beautifully to the experiment, to the theory. So we said, well, if this is indeed true, this is very simple. You have actin, you have motors, and you have a bilayer. Can we now reconstruct this in an artificial system from these very simple elements? And so Darius, Kabir, and Abrar, they set about to do this, and that's what they ended up doing. So this is a lipid that's present in a supported lipid bilayer. And here's myosin, and here's actin in 3 different colors. And now you can see when you add ATP, you make these fantastic clusters, which also begin to cluster the lipids there. In fact, what happens is that once the clustering happens, during the clustering, the lipids cluster, and once the clusters of these things stabilize, the lipids actually become declustered, saying it is the activity that is clustering these molecules and nothing else. So this was really exciting for us, and okay, so now many of you may have thought about this, but I'll answer the question. The transmembrane proteins can associate with the actin across the bilayer because they have actin binding motifs at the inner leaflet. What about GPI anchored molecules? Of course, as I mentioned when we started, GPI anchored molecules are molecules which have proteins that are anchored to the membrane via a glycolipid. Therefore, they cannot see the inner leaflet. There must be some connection between the outer leaflet and the inner leaflet that makes this connection, right? And the connection turned out to be solved when Ram and Ria and Ambika in my lab went to work with Ram, and they built a series of glycolipid analogs, painstakingly from synthetic, really tour de force of synthetic chemistry, and we found that it was only when you introduced long saturated lipid containing GPIs on the outer leaflet that you could actually get the clustering of these molecules taking place. So we needed saturated acyl chain containing GPIs at the outer leaflet, of course cholesterol dependent clustering, and that's what we required. And then we asked what was the clustering lipid agent at the inner leaflet. And the clustering agent at the inner leaflet turned out to be phosphatidylserine. Again, long saturated acyl chains. So you had long saturated acyl chain containing lipids at the outer leaflet, long saturated acyl chain containing lipids at the inner leaflet, and of course there must be a connection between the inner and the outer leaflet, and now we know where that connection is. And that connected the inner and the outer leaflet, building a structure like this. So what can this do? And here again, Madan and his student Anirban did some really heroic asymmetric bilayer simulations. So here are GPI anchored proteins embedded in the outer leaflet, phosphatidylserine in the inner leaflet, in a membrane that is actually at the physiological temperature. There's nothing that's making it cluster, but if you cool this membrane down, you'll see clustering. But you can do one more thing. If you now simply hold the phosphatidylserine at the inner leaflet in one place by making them immobile, the GPI anchored proteins come for the ride. So I'll just show you that simulation again. So if we'd held it, the lipids were like this earlier, we held those lipids, they came together, and the GPIs came in for the ride. And this suggested to us that the membrane is doing something, the cell is doing something like this. It's collecting actomyosin at the inner leaflet, holding phosphatidylserine and building this little nano domain in the membrane. And because we had simulations, we could actually look at the architecture of the lipids there, and those were liquid ordered little nano domains that were being built at temperatures at which you wouldn't expect any kind of ordering of the membrane to take place. So obviously we said if the simulations are predicting this, we should go and ask ourselves whether in the cell, if you look at where these clusters are, we should also begin to see ordered domains in the membrane. And so we needed a probe to visualize these ordered domains, and Satyajit, working in the lab, he used a probe that is a solvatochromic probe called LORDAN, which was, has been developed over years and, used very successfully by their colleague Catherine, who sadly is not here anymore. But what Satyajit found is that you could now use the wavelength-sensitive imaging of these solvatochromic probes to ask whether you had liquid ordered regions or liquid disordered regions in the membrane by simply looking at what wavelength these fluorophores that are embedded in the membrane are fluorescing. If they're fluorescing in the 450 nanometer wavelength or they're fluorescing in the 500 nanometer wavelength, you can distinguish between these two types of domains. And what he found was that using our anisotropy measurements coupled to the measurement of what that property of solvatochromic probes is called generalized polarization, just to confuse all of you, because we are looking at fluorescence polarization. The solvatochromic probes are also described to be looking at generalized polarization, but don't get confused. Just look at the wavelength differences between the solvatochromic probes at ordered and disordered regions. So what we found was that at very low fluorescence anisotropy regions, where the GPIs are highly clustered, you have regions in the membrane where you have extremely blue-shifted anisotropy, generalized polarization, which is indicative of extremely high order. So now we have a picture where we are able to connect how the cell is building these ordered patches in the membrane. The cell is building these ordered patches in the membrane using what we think are the active mechanics of the actin, which build these little nanoclusters, and then it's the lateral interactions with lipids that would like to associate with these little nanodomains that then ends up in constructing these larger liquid ordered patches, which we now call active emulsions. And these active emulsions are likely to be the so-called membrane rafts that we've been sort of chasing after for many years. And what I'd like to show you now is what this means for the cell membrane. And of course, these patches that I've been showing you all along of deep blue patches of depolarized fluorescence and anisotropy are precisely those sorts of liquid ordered patches in the membrane that we are now able to visualize and correlate. So what I've told you so far is that the heterogeneity we are seeing in this kind of property of ordered and disordered lipids in the membrane is a consequence of the active mechanics of cortical actin, right? And any transmembrane protein with an actin binding domain can form clusters. Of course, those clusters are not going to be sensitive to membrane order or anything. They're just formed because they are interacting with this active dynamic actin. Lipids, on the other hand, are going to be driven to building these sorts of domains if they have the right chemistry and the right amount of cholesterol so that you can build these ordered patches, and you can build these ordered patches when the actin is being generated. So this now actually tells us that the surface is in fact a membrane actin cortex composite, and that this composite lives in a sea of lipids, which, presumably is the Singer and Nicolson model of the fluid mosaic. But that gives rise to more structures when that membrane is actually present in a living cell and interacting with the dynamic actin cortex. It is interacting with the actin cytoskeleton underneath, giving rise to these very specific lipid environments, which I would argue are what we have been calling membrane rafts for the last 30, 40 years. But now we know how the cell is building them. And it's building them literally brick by, or should I say twig by twig, along with the consumption of energy and ATP, because it's overriding the thermodynamic tendency for the membrane to be homogeneous. And then there has to be, and this is what we are very excited by, a third class of molecules, which are called the active molecules, which regulate all this. Now, because you're using ATP, all this is going to be subject to regulation. And we know from work, from, say, looking at cells that have, sensory systems like the integrin receptor, that these receptors are able to, in fact, serve as our prototypic active regulator. So the integrin, let me explain this for a few minutes. Who's keeping track of time? It's not yet 2 hours, is it? No. I have 10, 15 minutes. Yeah, okay. Anyway, time goes fast if you're having fun, but I thought it was almost 2 hours. No. Okay. So then I have plenty of time. All right. So now these integrin receptors, they sense ligand and receptor ligand types. So they're sensing the chemistry of the extracellular matrix. They're also sensing the mechanical stiffness or the mechanical environment, or the micromechanical environment of the cells that the cells live in, because of course these receptors are on the scale of a few nanometers as well. So what we discovered, in fact this is what Joey and Thomas were doing, they discovered that when you look at cells that are now plated on, these are cells that carry integrin receptors which can bind to fibronectin, which is a very common integrin ligand. The cells, when they arrive on a substrate, they look, and that's, again, we are looking at GPI anchored protein clustering. We could have also been looking at our LORDAN imaging for liquid ordered domains. We'd see the same thing. So what you see here is when the cell arrives on the surface, it is first extremely orange, and then it becomes extremely blue, which means that it is building clusters as it arrives on the membrane, and those clusters are generating ordered domains. So in fact, as an aside, using this assay that Joey developed, we discovered that this clustering is sensitive to the chemistry of the interaction of the integrin. Means it's highly specific, highly regulated. It only happens when integrins sense fibronectin. Certain types of integrin sense fibronectin, but it does not happen when integrin sense laminin or collagen, and it requires a mechanotransducer. So when the integrins are stretched or experience force, that a protein called vinculin is activated, and that is required to build these clusters. So you have nanoclusters or liquid ordered domains formed upon integrin ligation and mechanical activation. And it turns out that this itself is an extremely powerful way by which the cell does things, and what it does, and I mean, we could obviously say, well, it does this, and so what? Who cares about this? Well, it turns out that the cell cares. It cares quite a bit, because when cells are undergoing this sort of rapid expansion, the membrane is getting depolarized. The area is expanding very quickly, and then the area sort of, the rate of change of the area relaxes back, and the membrane actually relaxes back to some sort of a native clustering state. And it is at this state that the cells require these domains to be built. I mean, to tell you what these domains do and what they are going to be useful for will take another seminar. So I will probably not go into that. But I should add that the cells themselves use these liquid ordered domains after the engagement of integrin signaling and mechanical activation of vinculin to recruit effectors like RAC, so that those RAC molecules can get activated and allow the cell to start spreading. And they do this locally. And this is very exciting because where the integrin is activated, the RAC is recruited to those sites by the building of these domains. And so these domains, in some sense, if you think about it, are acting as integrators of chemical and mechanical input, which then provide the cell with the possibility of doing something at the next step. So they're integrating this information in the membrane. And of course, this can happen, within seconds of the integrins being engaged and activated. So there's no integration happening of the signaling happening far away from the membrane. It's happening all at the site of the membrane. And this is something that we're really excited by, and of course we want to begin to ask questions on what kind of RAC activation happens here and what kind of effectors are being produced. Now all this would be moot, and very specialized if there were no generality here. So I want to go back to evolution again and say that, these processes that I've just described to you are not specific for integrin receptors. They're not specific for the cells that we study. They're actually incredibly general. The evolutionary antecedents of what we see can be captured if you look at the prevalence of the various players, in addition to, of course, the prevalence of the lipids themselves across evolutionary time and space. And in fact, that's what some colleagues of mine working at the KITP Quantitative Bio workshop that, in fact, the rotation project that my postdoc Thomas and I had conducted, we went and looked for the evolutionary antecedents of these molecules, and we found that structures that were involved in maintaining the clusters and connectors are present across all eukaryotic phyla. From single-cell protists to multicellular organisms, the entire ensemble has this. We found a blank space here, and some of those connect to certain kind of plants. And that is also very interesting, but it's a subject for another talk. So what we have seen so far is extremely general. And what is it that I've been trying to show you now? So I've been trying to show you that, and why is this so exciting? It's exciting, one, because it can build patches that integrate information, chemical, mechanical information, so that next steps or that a cell needs to engage with can happen. And I think this provides, especially for a cell during locomotion, the capacity to sense its next step, and therefore undergo taxis, which is sensitive to this surface. And we see, if we get rid of these domains, we can do that without affecting these molecular players simply by changing the lipids in the membrane. The cells are unable to sense these surfaces in any reasonable manner. Okay, so what else can this do? And I think this is a proposition that I'm going to leave you with, is that when the cells build these domains by active processes, not through passive thermodynamic means, but by active processes, they actually create lateral pressure differences of several atmospheres between this configuration of lipids and that configuration of lipids. So if a protein, say like an integrin receptor, unsuspecting molecule like this, sits in a liquid disordered patch, then finds itself in this ordered patch, it is going to get into a very different conformation. And these different conformations, so basically these different conformations, because they can be produced locally, will drive conformational change in membrane proteins, which are likely to have functional consequences. So, so we have also discovered a new solvatochromic probe, which will allow us to pick up these things in a more sort of real-time manner, where the dye is localized to the membrane, both the outer leaflet and the inner leaflet, by using some experimental tricks. And we can watch these patches of the membrane go from ordered to disordered in real time, and now see what can happen to the proteins in those regions. So I'm going to stop here by saying that this capacity for these active molecules, especially if they happen to be signaling receptors, the capacity for them to sculpt their local membrane environment is probably going to be important for a vast range of molecules, simply because integrins, I've already shown you, do this. GPCRs, receptor tyrosine kinases, and other sorts of molecules, all of them activate the same downstream signaling machinery that's necessary for producing the actin and also for producing these domains. And so we think there's a cycle of both activation and feedback from the membrane that regulates the structure and functioning of these molecules. So let me end by saying I hope I've given you a tour of how we are thinking about the structure and organization of cell membranes, and how we've moved from thinking about the membrane as a fluid mosaic to these thermodynamically driven rafts, to now a much more active picture of active membrane composites, driven by these multiple interactions with our physicist colleagues, and I'm actually very, quite excited by the prospects of what we will be finding next. So, so in conclusion, just to spell it out, when physics meets biology at the cell surface, they still continue to reflect an intertwined relationship between these concepts that have been borrowed across the disciplines. And just leave you with this idea that the edge of a living cell is a living, breathing skin, and it's mechaноchemically regulated by this active membrane composite that allows the capacity for orchestration, self-organization, with consequences for cellular function and behavior. Okay, so I'm going to stop and see if there are questions. Thank you very much for your time.
As earlier. But what is the interest, how nature, or just like you have synthesized phosphatidylserine, why nature has selected this, or there is any other alternate available for nature to this? And most of the place you are talking about the patches. Basically this is all the interaction of the molecule, and these all interactions should be non-covalent interactions.
Correct. So your two questions are, why has nature selected this specific chemistry? So the chemistry comes from systems that existed earlier, which is the bacteria, right? Bacteria or eukarya. They don't have, at least so far as we know, they don't seem to have this kind of an active composite machinery. So the machinery then comes along for the ride, if we map it through evolutionary time, and converges into the last common ancestor of the eukaryote, where the machinery exists along with the chemistry, and so now the cells can use it. So I mean that's at least my simple-minded way of thinking about this in terms of what selected what. I mean I think they were independently generated, and then they come together in and provide new functionality. The second question, yeah, you're right. These are all non-covalent interactions, And they are the standard interactions one might expect from lipids interacting with cholesterol and creating these sorts of patches. But now, driven by activity, they can be realized in a regulated manner. Right? They're the same, potentially the same interactions that exist in artificial membranes at lower temperatures when phase segregation starts to begin.
Yeah, Jitu here.
Yeah. Who?
Here.
Yeah, hi.
Hi.
Yeah. Hi,
So
Raj.
fantastic tour of membrane biology. What I wanted to ask is regarding the general understanding that people have that you actually have certain self-organization of the lipid components before the actin comes in, correct? Or the understanding that I get from your talk currently is that there is absolutely no organization?
I would argue two things. Yeah. Of course, there would be fluctuations of composition depending on the temperature and the composition of the material, the material composition of the lipids. The fluctuations in composition will be there because it's a heterogeneous mixture. Now, but there will be no so-called phase being maintained or formed simply because the membrane is maintained at a temperature higher than the melting temperature of that phase. If you actually take this membrane in a living cell and remove it from its cytoskeleton and then start to cool it down, you will see phase segregation happening, right? So it depends on what you do to the membrane. But at the temperature at which the cell lives, you don't see this composition, segregation, and it only occurs when it interacts with this active machinery. So, but then, of course, because you've begun the interaction, more interactions start coming into play, and you build literally a localized mesoscopic domain, which is an ordered patch. And those require interactions that are being motivated by what's in the membrane.
So basically, the lipids are, doing a passive role in this whole process.
But they are,
they are required, but they are actually doing a passive role in terms of the organization as such. Is that the general understanding that we are coming to?
Right. The proteins are driving this kind of a domain.
Interesting. Yeah. Thank you.
Hello. Jitu.
Yeah.
Beautiful presentation. So I have two kind of related questions. One is how specific is this general clustering? I mean, let's say if there are two receptors which bind to different ligands but interact with actin.
Right.
Is there a chance that these two receptors cluster together?
Actually, that's the PhD thesis of Abrar. What he found is that if you take two types of actin binding motifs which have identical binding affinity for actin, you can bring disparate molecules that have the same actin binding domain together. But if they have different actin binding motifs with different affinities, you actually sort them. You sort one cluster from the other. So if the affinity of the interaction between the actin and this dynamic actin and the dynamic actin and the molecule in question is able to sort based on this activity.
So, I believe the non-lipid part of the receptor, which is extracellular domain, might be contributing to specificity of this clustering as well.
It certainly can, yes.
Okay.
So, but for us to ask specific questions about what is the actin doing, we can eliminate the outside of the receptor. But of course, the receptor will have homomeric interactions amongst itself or heteromeric interactions, and all of those things will play a role, but the actin will modulate them.
So the question I have is, do you think there are similar clustering in the internal membranes, for example, ER or lysosomes and all those?
Well, I would imagine there are, certainly for membrane proteins.
Right.
For the lipids themselves, the transbilayer connections may become somewhat more difficult unless you have lipids that are able to stabilize the low concentrations of cholesterol that is present in these inside membranes. So the composition changes, you see, dramatically from inside to outside. You have very low cholesterol in the ER, very high cholesterol in the plasma membrane. And so perhaps you need different lipid chemistries to do that sort of thing. For the lipids, but for proteins, definitely.
Yeah, so lovely talk, Jeetu. I was wondering, do you have a sense, because these are short actin filaments with their associated non-muscle myosins, is there a separate pool of actomyosin conglomerates that are maintained membrane proximal and that they don't mix with the general pool of sort of actin which would be involved in stress fibers or other actin structures?
Right. Yeah. So what I didn't go into is this whole zoo of what's happening to the actin. The actin, the cortical actin near the membrane, we think has two flavors. One is this cortical filaments which are long and filamentous, which are suffused with non-muscle myosin 2. And now it's this cortex, when it flexes, that proteins that can bind to this cortex, the actin in the cortex, undergo this clustering behavior. But the lipids, I'd have to say, have a separate and distinct actomyosin machinery that is producing what is happening to the lipids. And there you have a different class of myosins, myosin 1, which binds to the lipids directly and recruits short filaments locally. And we know that these short filaments are actually made, short filaments and long filaments that are doing this are made by formins. So we know that. Yeah.
Great talk. I'm here. Yeah.
Hi.
Okay,
right.
hi.
Yeah.
So I was just, from your talk, you say that these interactions and this organization essentially happens in eukaryotes. And in bacteria,
right,
what happens? I mean, you have membranes. You also have, homologs of cytoskeleton proteins. You don't have cholesterol, but what is the what is the information on the organization and the dynamics of bacterial membranes?
Right. So I think in bacterial membranes, there are nascent. There are evolutionary related, structures which are so-called actin and microtubule-like or tubulin-like. But there are no known motor drivers. But I'd imagine that there are enzymes that are driving, the movement of particles in the membrane of a bacteria, which can also do the same thing. Or there are molecules that can patch lipids and sort of do passive patching. And people are beginning to see these sort of things.
So this kind of organization without, if you just probe the organization itself, of the membranes, do you see similar kinds of...
Actually, you do, which is sort of intriguing. But I have a feeling that the organization in bacteria may be more not driven by activity as much as by proteins patching in one region, creating domains that are of distinct composition. That can also happen in a eukaryotic membrane, because you can patch from the outside and you'll build, structures also locally.
But the dynamics of that you would expect would be, it's not known, is it?
Would be on the dynamics of the turnover of those patching components.
So this would be much slower than in that case.
Jeetu.
Hi, Amit. Yeah.
Great journey. Good to see that. Just a couple of small points. In response to Shankar's question, you said if you take the membrane out of the cytoskeleton and cool it, it will separate into the domains. Right. Yes and no, because unless you separate the proteins, because proteins also a broaden phase transition, right? We know that from early experience. I'm sure you meant that. And the second thing about, somebody asked about interleaflet interactions. There are cases, and you talked about low cholesterol in here. We showed long number of years back, cholesterol does form some kind of dimer. And recently we did some simulation, not published yet. We showed they have sufficient time scale in physiological domain. Okay, and this Sushmita and my work started, but then we did with other, and Manuel Priyat also showed that in some membranes. So there could be lipid crosstalk in transbilia. In fact, that has been shown in some explanation of raft also talks about it. In fact, interdigitalization of proteins is found under physiological conditions like what we did last night. there was a party, we drank some alcohol. Okay, that happens. So maybe that's also should be considered.
Yeah, so I agree with you, but I would just say that I think the idea that you can get segregation in the absence of doing something, segregation of lipids in the absence of doing something, will be very...
No. That's,
you have to do something.
Sure.
And
that adds up. Yes,
that adds up. Yeah,
sure.
but you
Absolutely.
need the composition, and and one area that I think is incredibly interesting is how is compositional homeostasis of these membrane components maintained? Right? We have no idea.
Yeah.
I mean, you take the membrane of a ES cell, or you take the membrane of a fibroblast, of a differentiated fibroblast, they're completely different. But if you take it today, or you take it next year, or you look at it last year, the ES cell looks like an ES cell, the fibroblast looks like a fibroblast.
Absolutely.
And that compositional control is something that's really very, very important. I think very important, and we need to understand how that happens.
Thank you.
So I think there are student questions from students at the back. Yeah.
Yeah, a fascinating talk, sir. What I was really interested about to know is, the edge of a cell,
right?
The architecture of the network assembly that we find there really dictates how the edge of the cell looks like. For example, the protrusive forces that are generated, forming lamellopodia, filopodia, etc. Now, if you look at it in the context of the kind of actomyosin clusters that mediate the kind of domains that you see, is there a relay of information from these clusters that are produced to bring about the formation of these network assemblies required for these formations of these really specific functional
right. No, I was sort of briefly mentioned that when I was talking to Jyotsna, that if when you build these clusters with the engagement of the integrins, right? That's a very local interaction with the ligand of the integrin and the mechanical nature of the surface. And that local interaction creates a local domain where the assembly factors for the lamellopodia are recruited. That domain recruits this, the possibility of maintaining an active rack. And now, at that site, you will begin to build a protrusion, and not anywhere else, right? So you're right. I mean, this information is now transmitted into a structural remodeling of the membrane by this kind of a mechanism.
Also, are the kind of actin clusters that you find between connectors and integrins, for example, is it the same kind of clusters that you see? The actomyosin clusters that you find, is the architecture of that similar?
Between what and what?
Okay, the interaction between transmembrane domains with actomyosin clusters and integrin proteins interacting with certain...
So integrins actually, do so many things. They activate the actomyosin machinery, which then leads to the flexing of the cortex. It also leads to the building of specific stress fibers, which operate through another machinery. Those stress fibers then bind to the integrins, and they reinforce the integrins, and all sorts of things happen. So once one starts looking, you see so many things that these systems do that one needs to look at them perhaps all together and understand what's unfurling in front of you. But you can now begin to see the cascade of events as they happen.
Thank you.
Hi. Fantastic talk, sir.
Excuse me, that would be the last question for this session. Yeah.
All right.
Thank you. So we have an interactive session again. All right. So I think you can save your questions, then we can take those questions later. So we'd like to thank you with the memento, and I would like to call Dr. Vinay Nandikori for felicitating.
Thank you. Okay.
I have not heard you talk about all your work at a stretch.
This is
pieces, yes.
This is part of it. Wow. Wow. That's my favorite color.
Yeah.
Oh, wow. Thank you.
T-shirt.
Oh, fantastic. Yeah. Amit, I said it has to be one size smaller than yours. Whatever you take.
Thank you, sir.
Thank you.
I would request both of you to stay back.